Hi guys, welcome to section B of chapter 3. We're going to talk all about protein synthesis today, which is the production of proteins. The proteins are produced using the code from that DNA that we've been learning about, okay? So there's really two processes, like two steps of protein synthesis that we're going to focus on. The first step is transcription. When you transcribe something, that basically means you're copying it down, okay? The second step is called translation. When you translate something, you like interpret it and change it into something else. You almost like decode it, okay, which is exactly what's happening. So we'll talk about both of those steps pretty detailed here. The overall idea of protein synthesis is just that new proteins are being made. To synthesize something means to make it. And they're being made from that code found within those strings of letters of the DNA, okay? So the first part is transcription. Totally takes place in the nucleus. That's where the DNA is. And a readable copy of that code is made within the nucleus. That copy eventually leaves the nucleus for translation, but right now we're still inside the nucleus for transcription, okay? So here are the steps, okay? This special enzyme comes along. Enzyme is called RNA polymerase, right here. A lot of enzymes end in that ASE suffix there. So RNA polymerase comes in and it takes apart that double helix of the DNA, kind of separates those rungs of the ladder, okay? Then a single strand of messenger RNA is produced from the DNA. So this right here, you can see the RNA right there. It's actually called mRNA, and that stands for messenger RNA. It's the copy. This is the readable copy that's going to leave the nucleus, okay? Now there's one big difference. RNA, by the way, when we have this R, this is another nucleic acid, but it's now ribonucleic acid. DNA is deoxyribonucleic acid. RNA is just ribonucleic acid, so it's a little different, okay? There's a big difference in the bases, though, if you notice. This U for uracil is replacing any of the T's for thymine, okay? So if you look at the string of RNA up there, anywhere you see an A on the DNA, you would expect to match up with a T. It's being replaced with a U. There are no T's. There's no thymine in the RNA string at all. I really don't know why. That's just how it is, okay? But look at these bases. They're still, these base pairs, I mean, they still are following a pattern, okay? There's an A and T. It's okay to have the A in RNA, G and C. Oh, there's a U instead of a T, but they still follow that same pairing, okay? So then this piece of messenger RNA is going to take off, and then we're going to talk about translation, okay? So we're continuing the process. Translation is the conversion of that piece of RNA into an actual protein, okay? And that takes place in the cytoplasm of the cell. And it uses the organelle called a ribosome. It's little tiny dots, okay? Here are the steps. So the messenger RNA flies out and it attaches to a ribosome. This big blob right here is the ribosome, okay? And then here's the messenger RNA. And that messenger RNA literally kind of travels through it, okay? Every 3 letters is a code. So every set of 3 letters is called a codon, and it codes for a specific amino acid. Amino acids are the building blocks of proteins. So here's a single amino acid right here. Those all get put together into this chain, and that's the protein, okay? You can see that we've got this thing called transfer RNA. So the tRNA, it's got its own 3 letters on the bottom of it. Those come and match up with the codon. Follows the same rules. G's and C's go together, A's and U's go together, okay? Or A's and T's, okay? If the T is on the, oh, never mind, no T's because we're in messenger RNA. No T's at all. So just A's and U's and G's and C's, okay? The transfer RNA with this 3-letter code that matches with this 3-letter code has a specific amino acid. this matches with this, and it's a different amino acid. Okay, so that's it. This is how the code is decoded. Those transfer RNAs bring in the 3-piece matching codon. It's actually called an anticodon. And then those amino acids all get stuck together with another enzyme. And then when it's all done, I stepped ahead. There's the anticodon. These are the anticodons right now. I explained it before I got you your notes. Sorry. Matches with that codon on the messenger RNA. But when it's all done, a special codon comes in and it signals stop. Okay. So there's a 3-letter code. I don't know what it is. It's at the very tail end of the messenger RNA, and it's anticodon that reads that doesn't have an amino acid on it. So kind of a blank transfer RNA comes in, and then that allows this chain to break off and go off and be transported through the cell to do its job. Okay. So let's kind of look at this all together. This is our nucleus right here. Okay. So inside of the nucleus, that's transcription. You've got that DNA opening up right here, and the messenger RNA making its copy. Okay, and then it leaves, goes out into the cytoplasm. So now we're talking about translation. And you can see it might be a pretty long string. This string of messenger RNA is attached to this ribosome, making that protein, but it's going through this one and making that protein. It's going through this one and making that protein. You can get multiple copies of a protein off of the same piece of messenger RNA. So this is happening over and over, okay? And then you just have a blow-up version of it. That's what's happening on the ribosome. We've got our 3-letter codons on the messenger RNA, and then we've got our 3-letter anticodons on the transfer RNA that bring in the specific amino acids, okay? So just so you know, kind of a fun fact, there are actually 20 amino acids in our bodies. We have 26 letters in the alphabet, and you can think of how many words we can make, okay? We can make all kinds of words with those different combinations of the 26 letters. So with 20 amino acids, there's a lot of different words you can make. There's a lot of different combinations of those amino acids to make a multiple selection of different proteins. But it's all determined by that code on the messenger RNA that comes from our DNA. Wild, crazy. This is happening in every single one of our cells all the time without us thinking about it, because God is such an amazing creator and such an amazing designer. This did not evolve from nothing. How do I know? Because of this. Remember I talked to you guys about the mousetrap the other day? There's your definition for RNA. You can get that copied down. So this just supports the idea of this being an engineered, planned, designed system, okay? We call that designer God. This concept of irreducible complexity. If you take one piece of this mousetrap out, it doesn't work, okay? Same with our cells. If we take one piece of this process out, it doesn't work, okay? But here's kind of a wild thing. Let me get back to where I was, okay? The whole process of protein synthesis, it relies on a protein. Ribosomes themselves are proteins. And so if proteins evolved by themselves and are already kind of randomly came into being, we don't know if it was a ribosome or not. Could have been any protein. I don't think that it happened at all, but my point is this system of creating proteins is an example of irreducible complexity. If we take a single protein away, it doesn't work. If we take some of the DNA away, if we take the RNA away, the fact that proteins are needed for protein synthesis tells you that somebody designed it and set it into motion. The same idea of cells coming from other cells, evolution doesn't explain where the first cell came from. It points towards God. So that is all I've got for you guys today. Hope it makes sense. See you later.